I'm going to be talking about some work done by one of my graduate students, Ben Tribble, and it's a project that he's been working on for a couple of years now. The work in my lab is really done at three levels, and traditionally I've been involved in development of vaccines and diagnostics. Also, you've heard a lot about the Host Genetics Consortium. And I think another area that really we need to begin to work on is in the area of nutrition, and not just on understanding how PRRS affects growth and things such as feed efficiency, but then beginning to look at the effect of PRRS infection at the level of the microbiome. This whole strategy is not designed to hit a home run. Finding genes involved in resistance is not designed to make a perfectly resistant pig. Collectively, these strategies create an opportunity to begin to take back what PRRS takes from us. In other words, if you see an overall 10% reduction in growth efficiency as a result of PRRS infection, we can get some of that back with better genetics, combined with better vaccines, combined with better nutrition. So if anything, there will be probably, in my lifetime, no cure for PRRS, but I think we can begin to manage it to the point that it becomes inconsequential. And so I'm going to talk about the PRRS Host Genetics Consortium, but I'm not going to talk about genetics. Infecting these pigs, large number of pigs experimentally, shows us the impact of PRRS at the population or the herd level. And if you just take and weigh pigs over time and you look at the distribution of weights at the end of 42 days, you can see that the population structure has pretty much fallen apart. You no longer see a single distribution, but you see a multimodal distribution in which you can find pigs that we call winners, but you can also find pigs that we call losers. And these black dots refer to just pigs that have not been infected with PRRS. And so one of the things we try to understand is, why are these PRRS-infected pigs different from those PRRS-infected pigs? Is it the genetics of the virus that is used for infection? Is the genetics of the pigs? Is it are there some other factors involved? And so just to kind of review a few points that everybody knows, PRRS is a persistent infection. It's persistent within a production system, very efficiently transmitted at every step. It's also persistent in the pig. And you saw this picture earlier several times from Jack, in which viremia doesn't go away. It keeps coming back. And if you can imagine that a PRRS-infected pig in the nursery, you will continue to see waves of reactivation of viremia all the way into the finisher. But each wave is just slightly dampened, but the virus is still there. And so we've also seen versions of this slide in which during PRRS infection you see early suppression of innate responses. You do see the production of antibody, a fairly robust antibody response, and eventually you do see viremia disappear with the initiation of neutralizing antibody. However, this is generally delayed and suppressed. And again, we see this prolonged viremia, which we have called persistence. And so another aspect of this is the fact that PRRS possesses a very complex virion structure with multiple viral proteins that are interacting with multiple receptors. And this is really the current model up until the time that Randy Prather constructed his silyl adhesive knockout pigs. And this is a very convenient model because now we focus a lot on GP2 and GP3 and GP4, and we can begin to maybe disregard GP5 as being important. But we're going to come back to GP5 at the end of this talk. The other aspect of PRRS immunity is the fact that the surface proteins are generally poorly immunogenic, with proteins such as the N protein and NSP2 as being immunodominant. And then finally, when you do find something that looks like an epitope or a neutralizing epitope, these epitopes are often found in regions that are hypervariable. In other words, with the idea that PRRS, through antigenic variation, can escape a neutralizing antibody. And then we all know that there are PRRS vaccines. We've had them since 1994. If you look at the history of the use of modified live vaccines, they have been demonstrated to increase profitability. They do have a benefit. However, there always have been several limitations noted, including this idea of incomplete immune protection, as well as the absence of a DIVA vaccine. We don't generally regard killed vaccines as being effective, and again, we still use acclimation as a means to develop some sort of herd protection. And to some of the challenges over the years that we've had to vaccine development is the fact that there are these N-glycosylation of the GPs, especially in regions that have neutralizing epitopes. We have these immunodominant decoy epitopes. There are hypervariable and hypervariable regions within these epitopes, and the infection itself alters B-cell repertoire development, as well as what you've seen with Dongwon and others, the subversion of early innate immune responses, which can affect adaptive immunity later on during infection. And so, but we do know that neutralizing antibody likely plays an important role. And this is the work done by Fernando back in 2002, saying that if prior to infection you add passive antibody, you can provide sterilizing immunity. And the work that we have done during the consortium, looking at large numbers of infected animals, you can actually see an inverse relationship between virus neutralizing titer and virus load. So not only is neutralizing antibody important for providing protection as a result of vaccination, it also plays a role during clearance. And so the antibody response during PRRS infection, PRRS neutralizing response is weak and delayed. That's generally accepted. And that neutralizing activity is primarily directed against the homologous virus. And one of the questions that we asked in infecting these large numbers of pigs, can we find pigs within these experimentally infected populations that can produce both a strong and what we call a broadly neutralizing antibody response? And if so, what do these pigs see on the proteome? What are the epitopes they recognize? And we believe that that relates to the genetics of the virus. And then the second question is why do pigs see it? Why do they produce this response? And we believe that that really relates to the genetics of the pig. And so you've seen this before, our standard host genetics consortium nursery model. This is the one that both Jack and Joan have shown you before. In this case, these are pigs that are vaccinated. They're challenged with virus, and then 42 days after challenge, they're terminated. And again, one feature of this is that there are 20 people in my lab who do nothing but bleed pigs, and they bleed them frequently. And so the virus neutralization assay that we used in this study is a standard assay, making serial 1 to 2 dilutions of serum and incubate those with approximately 200 TCID50 of PRRS. Incubate them for 4 days, and we use CPE as the endpoint. And then with each plate we include positive and negative controls, as well as the back titration of the virus. And so when we go back and we ask this question, well, why are we interested in broadly neutralizing antibody? And this really originates from 10 years of studying HIV patients who produce antibodies to a broad range of HIV isolates, and they never go on to develop AIDS. And some of the features of these broadly neutralizing antibodies is that they generally recognize conserved epitopes and are capable of neutralizing genetically diverse isolates. These conserved epitopes can be both conformational or they can be small linear epitopes. However, one interesting feature about epitopes recognized by broadly neutralizing antibodies is the fact that oftentimes they're near areas of glycan shielding, or they're near areas of hypervariable regions. And with the idea, obviously, glycans can block the recognition of epitopes, but the presence of these hypervariable regions may be likely sufficient to, when you have a mutation, that it is sufficient to change the conformation enough that you lose recognition by antibody, but you still retain function of the protein. And so in our study with the consortium pigs, sure enough, if you vaccinate a pig, and we use in this case, we use a heterologous PRRS challenge. If you vaccinate a pig, yeah, you can see protection. You can see reduced viremia. You can see reduced clinical signs. In this case, we were fortunate enough to have pigs with blue ear. And even though this is not pathognomonic for PRRS, oftentimes it's just a convenient marker for identifying a PRRS-associated clinical sign. And then when you pull a group of pigs at day 11 and you look at lung pathology, in all the vaccinated pigs, the lung pathology is decreased. Now, one of the things that we're developing, because we have so many pigs, is really a photo scoring technique in which we photograph two students in my lab who are both DVMs, are photographing lungs and actually digitizing the images and then making histopathology or gross histopathology evaluations. This is a convenient way to save the data. Plus, you could have several pathologists look at the blinded digitized images and make their own conclusions. And so when we talk about, well, how do we assess this broadly neutralizing activity, we perform neutralizing assays, in this case, with 7 different isolates, including Lilystad virus, which is a type 1 virus. The viruses that we use, this shows amino acids at differences in GP5. This shows nucleotide differences in ORS 2 through 6, in which case the lowest identity is for LV, 65%, and the highest, greatest is about 98%. This is just a phylogenetic tree showing the isolates that we used for neutralization, how they're distributed. And so based on neutralizing responses to this panel of viruses, we can take our sera from these PHGC studies and we can group them into 4 groups. One is pigs that show no virus neutralizing activity, pigs that only show homologous activity against the virus used for infection, heterologous, in which case we say that they can neutralize several type 2 viruses, but not type 1. And then finally, we define broad in our study that they neutralize all the type 2 viruses in our panel, including the type 1. And so based on this, in a study beginning with 200 pigs, 100 vaccinated and 100 not vaccinated, we can build these distributions based on placing pigs in these 4 groups. And what you can see is that, yeah, vaccine does work. We move from a more homologous response to one that is heterologous, including one animal among this whole group that showed this broadly neutralizing response. The other thing that's interesting about this is that 13% of pigs, regardless of whether or not they've been vaccinated or not vaccinated, failed to develop any virus neutralizing response. And so out of these populations of pigs, we just pick pigs that show some unique neutralizing responses. This is an example of a pig in which we say the response is homologous. The titer, and this is log base 2, is huge. This is about a dilution of 1 to 1,000 neutralization titer. That's pretty good for PRRS, considering that when you have neutralization titers of 32 or 64, those are pretty good. But you can see is that even though this serum has a very high neutralizing, homologous neutralizing response, it fails to neutralize any of our viruses in our panel. And to contrast this against the one pig, pig 1645, that produced this broadly neutralizing response, you can find that, yes, you can still see this homologous recognition, but then you can also detect neutralizing activity against other viruses within the panel. This is an example of a pig that showed no neutralizing activity against any of the viruses. And then finally, in a separate PHGC study, which involved a different virus that was broadly neutralizing, you can see that this virus also is broadly neutralizing within this panel of viruses. And so one of the interesting aspects of this, and this is work that was just published out of Rowland, is that when you look at where does this antibody come from in terms of virus replication, and generally speaking is that those pigs that produce a homologous response generally show high peak viremia with clearance, but later on they show this reactivation or this rebound effect in terms of renewed virus replication. Whereas those pigs which produce more of a broad neutralizing response, they come from pigs that show more of a persistent low-level form of replication. Presumably the presence of the virus in the pig for a much longer time creates the opportunity to recruit more B cell clones as well as increase affinity maturation over time. Now, one thing is when we look at this rebound virus and we ask the question, well, what is it? It's actually a virus that has escaped homologous antibody, but is still sensitive to broadly neutralizing antibody. And so we can take these unique sera and we can actually select for viruses that are become resistant to neutralization. And we do this by diluting antibody against virus on a 24-well plate of Mark 145 cells. And this is different concentrations. This is just a checkerboard titration. We pick the last wells that show some CPE. We then bulk passage those and grow those up on a T25 and then resume the selection process. Eventually, after about 6 rounds of selection, we end with a virus that is resistant to neutralizing antibody. So these are really some of our results, and we see a couple of things going on. We can find mutations. For example, this virus we call K62R4, which was selected using our broadly neutralizing antibody 1645. We find a single mutation in the structural genes, and that's a deletion of amino acid in the M protein. Whereas others, it's a little more complex. And some of these mutations we know are not the result of escape from neutralization, but are probably the result of the virus adapting to growth in mark cells. Because remember, we're doing several rounds of passaging when we do this selection process. So what I want to do is just focus on this virus here, this Y10 deletion. It's very interesting because it is a deletion. So the probability that you're going to have a reversion is going to be low when we use this for further experiments. And so this is the location of this tyrosine that's deleted in our resistant virus. It's at position 10. The M protein is one of the most conserved structural proteins. This position 10 is kind of interesting because there is some variation between isolates. If you look at kind of a representation of the heterodimer between M and GP5, this tyrosine at position 10 is located right next to a serine that forms a disulfide bond with GP5. Also here we have the glycans represented as well as hypervariable regions 1, hypervariable regions 2. And it's Fernando who identified a conserved epitope, a linear epitope in this region. And so what we find is that this, our KS62R, or our resistant viruses, sure enough, is resistant to broadly neutralizing antibody, whether it's our 1645 or from another virus, 15-4. However, what's interesting is that this virus is still sensitive to homologous antibody. And so as one of the applications of using infectious clones, as Jay referred to, is that we took an infectious clone that also had a GFP marker, and we made a mutation and deleted tyrosine 10 in the M protein. And sure enough, our infectious clone behaved in the same way as the wild-type parent virus. It was sensitive or resistant to broadly neutralizing antibody, but still sensitive to homologous. And so what we conclude from this is that the resistance of this virus to broadly neutralizing sera confirms this role of tyrosine 10 in a neutralizing epitope. And so when we look at the location of this and some other mutations that we find, remember, we started out with the premise that GP5 doesn't matter because we already eliminated a role for sialadhesin. And yet several of the mutations we find come back to GP5. It's kind of like Godfather Episode 3, in which the more you try to run from GP5, the more it sucks you back in. And so what we've found so far is this deletion of a broadly neutralizing antibody, resistance to broadly neutralizing antibody in M. Now we've also done the same experiment with homologous, some of these unique homologous here, and so far the mutations that we find actually map to this region in the ectodomain of GP5. And so this really creates some really kind of interesting ideas about this conserved epitope here. We know that it's shielded based on these glycans, and we also predict that it's going to be protected based on these flanking hypervariable regions. A third way in which you can protect a neutralizing epitope is probably this M protein, in which mutations in here may change this epitope just slightly enough to escape broadly neutralizing antibody, and yet still maintain GP5 function. Another possibility we're looking at is that this is probably maybe part of a larger conformational epitope. And so some of the summary and conclusions: neutralizing antibody participates in the control and clearance of PRRS. There's a wide variation in the NAB response of pigs. Not all neutralizing antibodies are equal. And we can take these neutralizing antibodies and we can divide them into distinct groups. Virus rebound is escape from homologous neutralizing antibody, and then different neutralizing antibodies versus homologous versus broad recognize different epitopes. And then again, to kind of close this out in terms of genetics, is that understanding the virus and host genetics of broadly neutralizing antibody will be important for the next generation of vaccines. And so one possible implication in terms of vaccine development is that we like vaccines that replicate to high level, induce neutralizing antibody very quickly, and then are cleared. However, in the case of PRRS, we believe that this scenario really stimulates a homologous response. Whereas if you could develop a vaccine which is more persistent and maybe percolates for a longer period of time, recruiting more B-cell clones, undergoing affinity maturation, maybe this will favor antibodies that are more broadly neutralizing. So you really look at the strength of the neutralizing response versus whether or not you want breadth of protection. Just to finish up, I've said a lot about B cells, but not much about T cells. And Catherine Ewan in our group at K-State has been doing a lot with our SCID pig that we actually discovered as part of the host genetics consortium work. And these pigs lack B and T cells. One of the things we've been interested in doing is reconstituting these pigs. And we can reconstitute these pigs with bone marrow, and now we're actually beginning to reconstitute these pigs with only T cells, so that maybe we can look at the influence of T cells on PRRS immunity. And this is just an example, and this is just very recent data in which you can see the beginnings of a follicle that are forming in a lymph node of a SCID that has been reconstituted with T cells.